This is Unit 2, the first PowerPoint Part B, so the last portion of the first section. So enzymes are regulated in many different manners, and this was mentioned in your outline as well as partly in the first portion of Unit 2. Constitutive enzymes are the ones that are on all of the time. So 60% of the enzymes within bacteria are constitutive, which again means that they are never turned off. And again, that is 60%. Then the remaining 40% are actually regulated, which means they're controlled at both the enzyme and the DNA level. So what we want to talk about in this PowerPoint is how do bacteria regulate or control their enzymes? We already know about how enzymes work with the active site and the substrate and the different metabolic pathways that yield an end product. And this is controlled enzymatically through feedback inhibition. Remember that that works in a both competitive and non-competitive manner and is also effective and efficient in saving energy because once enough end product is made, the feedback inhibition stops the cycle from continually making more end product. Bacteria have a unique grouping to their genes, and they're known as operons. In the beginning of the class, we talked about how prokaryotic cells are metabolically quicker and the fact that they're so much slower and that they don't have membrane-bound organelles. Well, it's also unique to bacteria to have an operon. And again, what happens here is when you talk about transcription, things are transcribed or genes as a group, not individually. So when you go from DNA replication to transcription, that is regulation at the DNA level. And the major or big differences between prokaryotic and eukaryotic is in fact this gene arrangement, which in bacteria is referred to as an operon. So things are done in groups as opposed to being done one at a time. So the general overview of regulation at the DNA level has to do with how an operon functions, and then it's also regulated by proteins known as a repressor. So let's take a look at this particular slide. The picture up top as well as on the bottom are exactly the same. They are just two different places where the pictures came from. So an operon consists of three different portions. There is first the promoter, second the operator, and three the structural genes. This pink structure right here is known as a repressor. A repressor is constantly being made, and notice that it actually binds specifically to the operator. And the operator is one of three portions of the operon as a whole. In this case, we are talking about RNA polymerase, which synthesizes RNA because, again, the name polymerase. So notice in this picture on the bottom, the repressor is actually blue, not pink. As mentioned above, the repressor is constantly bound to the operator, and therefore the RNA polymerase is essentially blocked and is never able to transcribe these particular structural genes. Repressors are used in two different types of gene regulation, and we are only going to talk about induction. Remember this before test time comes. And the example that we're going to talk about for induction operons is the lac operon. Lac is short for lactose. So if you take a look at this particular picture, it's similar to the one we just discussed. In a normal status, remember that not only is the repressor constantly made, it's always present, but it binds to the operator. And you can see it right here, that it is bound to the operator, right here. So what happens is the transcription of these structural genes is completely blocked. I want you to acknowledge these glucose molecules throughout the entire environment. This is happening because glucose is available. That's why the repressor is constantly made. That's why the repressor is bound to the operator. And again, what happens is transcription is blocked. The system of the lac operon starts off in a repressed state. This particular picture definitely varies from the previous one because, again, we have our promoter, we have our operator, And here, this 1, 2, and 3, these are our structural genes. Again, in a normal situation, when glucose, which is right here, this green ring, when glucose is present, the repressor is constantly made, and it would be bound to the operator right here, which would then block transcription. But in this particular case, that isn't what's happening, because if you take a look at the picture, there's very little glucose present. But in fact, you see these lactose molecules present. We'll just focus on lactose for now. It's important that you understand that glucose and lactose are inversely proportional. So in simplified terms, that means when glucose levels are high, lactose levels are low. This is the normal situation. This is when the repressor is constantly being made, binds itself to the operator, and stops the transcription of the structural genes. But in this particular picture below, glucose levels are low. Notice there's only one glucose in this picture, and lactose has increased. This is a very different situation, and in this case, the repressor is being kept busy. In essence, the lactose is acting as a competitive inhibitor. Think back to when we talked about enzymatic pathways, and when the lactose is acting as a competitive inhibitor, it inactivates the repressor, and therefore it's not able to bind itself to the operator. So by not being able to bind to the operator, these 3 structural genes are in fact transcribed, and here is the tail of the mRNA that is growing. So I'm intentionally going to go back a moment so that we can go through this one more time. In the lac operon, what's unique is that an operon consists of 3 portions, which are the promoter, the operator, and the structural genes. In a normal situation, glucose is present in high numbers, and when glucose is present, repressor is constantly being made. That's the pink repressor right down here. And what it does is it binds itself to the operator, which blocks the RNA polymerase from transcribing these structural genes. This is the regular normal status. But glucose and lactose are inversely proportional, and therefore when glucose levels drop, lactose levels tend to increase. Lactose in this case acts as an competitive inhibitor. It keeps the repressor busy, stopping it from being able to block to the operator, and therefore this RNA polymerase, which would be somewhere right here, is able to go through and transcribe the structural genes. So further for regulation, the next question would be, what happens when there's a change in phenotype? How did it happen? What does it mean at the DNA and the protein level? So we are going to take a look at mutations. Mutations occur specifically in DNA. There are no conversations about mutations in RNA. This is specifically referring to DNA. There are 2 different types of mutations. One of them are spontaneous, and just like it sounds, it means the DNA polymerase has an error. So accidentally, if you look at the picture right here, guanine would pair with cytosine, and in this case, it happened to be a thymine, which is an error, and therefore this is spontaneous. The other kind are induced mutations, and for this, there are so many examples. When a woman goes to a dentist's office or to get a mammogram, the first question is whether or not they're pregnant, and then they throw the lead vest on because X-rays, UV, sunlight, cigarette smoking, all of these things are known to induce mutations. A couple more examples on this slide. We had already mentioned X-ray and UV light, which is radiation. So with UV, notice how cytosine pairs with guanine. That's normal. Again, cytosine and guanine, but in this case, the thymine should pair with the adenine, but that doesn't actually happen, and what ends up happening is the 2 thymines pair themselves together. This is known as a thymine dimer, di for 2, and it causes a kink within the DNA. This occurring one too many times, sunburn one too many times, it eventually alters the DNA to where then comes the concern of, let's say, skin cancer. So there are 4 mutations on this chart. This chart looks a little large. I'm going to move it up. But in actuality, they're not difficult to understand when you visualize what's actually occurring by looking at it. When you do your DNA homework, as well as when you look at something like this, codons are always 3 bases. So I always suggest to students that you start off by dividing everything into groups of 3. It makes it a little bit simpler to actually see what's happening. So I'm going to draw a couple of lines here. So the only thing I cut off on tops is that there are types of mutations. So please make sure that somewhere in front of you, you have an amino acid chart, because if you were to look all of these codons up, so groups of 3 bases, you would be able to see what protein they code for. So here you have a normal DNA strand. This is the parental strand. And now it's been transcribed into mRNA. Then you would use the chart to look up that UUU codes for phenylalanine. For example, GCA codes for alanine. So this is the normal polypeptide based on the amino acid chart. So now let's see what happens. The first codon, you have AAA, and here you have AAG. So you have, right here, a mutation. In other words, the DNA template had a mutation, which means when you transcribe it, instead of up top where the A pairs with a U, you now have a G, which is going to pair with a cytosine. So if you go to the chart and you look up the codon UUC, it codes for phenylalanine. But if you take a look above, that also coded for phenylalanine. When there's a mutation that the protein that's coded for is exactly the same as prior to the mutation, that's known as a silent mutation. That means there's no change in the amino acid sequence and therefore the protein that's been made through translation. Why is this possible? Because just like we have 26 letters in the alphabet and can make zillions of words out of it, many different codons code for the same amino acid. This is important biologically because if every single combination of letters or bases in a codon paired for something different, then basic spontaneous mutations would be deleterious or dangerous and cause issues all the time. So let's take a look at a second type of mutation. I'm going to go in and draw the lines again because it is simpler to visualize in 3. When you do your homework, you will see that you definitely want to do this. So look at the original normal strand, and right here it says CGT. Whereas right here, this has turned into CCT. So once that is transcribed, it's GGA. Originally, when it was CGT, it coded for alanine. Now, if you look up the codon GGA on the amino acid chart, it codes for glycine. This is known as a missense mutation. Missense means it codes for a slightly different amino acid sequence. It's only one protein, so it may or may not be a big deal in the scope of things, but essentially a missense means that it's just a slightly different amino acid sequence for one single codon during the translation step. So let's take a look at the third kind. And again, we're going to draw our lines. So here on the original one, this was ATA, and now it is ATT. So when that becomes transcribed into mRNA, we are looking up UAA as the codon, and when you look on the chart, it actually says that is a stop codon. So with a stop codon, is this bad? Well, it absolutely could be, or it could be no big deal. Envision 100 codons. If this type of, this is called nonsense mutation. If this stop codon were to show up at amino acid number 99 out of 100, essentially you've already transcribed and then translated the entire sequence, so it wouldn't be such a big deal. But if this were to occur sometime in the beginning of the structural gene, then it is in fact a problem. Or if the mutation occurred early on in the DNA strand and then transcribed and then translated, because then the rest of the entire polypeptide chain would not be made. So again, if there's a stop codon, this is known as a nonsense mutation. So now we're going to cover the last one, and frameshift mutations are called the most deleterious. Deleterious is another word for dangerous. These are the ones that impact most negatively what occurs in mutations. So we're going to draw our lines here for both of them because they are essentially the same. It's really just a matter of looking up the different codons when you have the amino acid chart in front of you. So look back to the original normal DNA right up top here. And you see here it's AAA, that matches. And right away, here's the ATA you see in the beginning, but something occurred called an insertion. An insertion means that a base was inserted. So now what happened? It shifted the entire reading frame. I'm going to do this in a different color so you can see it. We're going to do this in black. So right here in the beginning, you would have had AAA, and then ATA, and then CGT and GCA. Now, because you actually inserted this thymine, I'm going to do this in green, you have shifted the entire reading frame of this mutated DNA template strand. So a frame shift insertion is when you put in an extra base and it changes the reading frame. So then you can imagine a frame shift deletion is exactly the same thing. It's when you pull out a base or delete one and it happens to change the entire reading frame. Why is this the most dangerous and deleterious? Because every single thing that's read and after transcription and then translated, because this mutation originally occurs in DNA. So take a look at this picture. The strand gets mutated, then the mRNA is transcribed differently, and finally translation is different. So you have a major difference in the amino acid sequence. An example of a frame shift deletion is something that occurs with cigarette smoking. There could be deletions, there could be insertions, but ultimately these are induced from chemicals within the smoke. And again, for frame shift, there are two different kinds, deletion and insertion, and these in general are known as the most deleterious mutations that exist. One thing I want to point out and mention is something known as mismatch repair. So if you are typing a letter to somebody and you want to say happy birthday, and you start writing out happy and then birthday, and let's say you make a mistake. Obviously, the letter T is missing. So what would you do? If you were typing, you would hit the backspace button and delete the mistake. Then you'd go back in and continue to write what you were intending to write. DNA polymerase 3 has a unique ability to do something known as mismatch repair, which is no different than my example here of how a backspace works when you are typing up a paper. So here you have a mutated DNA because an incorrect base was put in. Traditionally, T, thymine, pairs with A, not a guanine. So with mismatch repair, what happens is an enzyme is going to come in and take out the incorrect sequence, and then again, DNA polymerase 3 is going to come in and fix the gap by repairing it and adding in the proper bases so that the complementary base pair is in fact working. This is an important mechanism, the mismatch repair, because we constantly have spontaneous mutations occurring. And if DNA polymerase 3 was not able to correct and make these repairs and put in the proper bases, then mutations would be more dangerous at a lower level and rate than they are today. So the last concept I would like to cover in this PowerPoint or this lecture is the Ames test. This is the final question on your DNA homework, and there's some terminology you need to be comfortable with, and then I'm going to explain how this test works. So the Ames test tests for something known as mutagenicity. Mutagenicity is the ability to cause mutations. Often mutations can become carcinogenic, which means cancer-causing, but it's very important to remember that something that is mutagenic is not always mean that it's carcinogenic. An example of that is caffeine is known to cause mutations in DNA, but that doesn't make it cancer-causing. Whereas in the other direction, if something is carcinogenic, it is definitely also mutagenic, because in order for something to be carcinogenic, which means cancer-causing, it implies that a mutation had to have occurred at some point. So we're going to talk about the Ames test, and again, the Ames test is to look for mutagenicity. Please make sure you understand that all mutagens are not carcinogens, so all mutations don't cause cancer. So the basic concept of the Ames test is that there are several strands of salmonella that are combined in a culture. And these particular strains of salmonella have pre-existing mutations that make them unable or not possible to synthesize histidine. A test is run and done in media that doesn't have histidine in it. So there are new mutations that can occur that can restore a bacteria's ability to synthesize histidine and grow in a media that doesn't have it if it causes a mutation. Therefore, if there's growth, you can assume that whatever is being tested or the suspected mutagen is in fact mutagenic. To summarize it, basically, what you have is when there's growth, and that implies that there has in fact been a mutation, only bacteria that are able to mutate to grow even though histidine is missing and required for growth, that's how we know that mutagenesis has occurred. So if you take a look at this picture right here, on one side you have an experimental tube, and on the other side you have the control tube. Both of them contain liver extract as a nutrient source, and again, both of them have a His minus salmonella strain. So again, that's a culture of salmonella that actually does not have the ability to synthesize histidine. The difference between the experimental and the control is whatever the suspected mutagen is. So we want to know if this particular product causes mutations or its mutagenicity level is added to the tube, whereas the control tube does not have that added. So this is then plated on medium that has no histidine. So not only does the plate not have histidine, but genotypically or phenotypically, salmonella in the culture is also His minus. At this point, the plates are incubated, and afterwards, notice that the control side on the right obviously had no growth. The reason there was no growth is there's no histidine being synthesized in the salmonella strain used in both cultures, and the media that it's plated on for incubation also has no histidine involved. Whereas on the other side, we did use a plate, or a plate was used that did not have histidine, but obviously whatever this suspected mutagen was able to revert the His minus in salmonella to produce histidine again and make it His positive. So phenotypically, that the suspected mutagen would be His positive. It essentially turned on the ability of this salmonella strain that normally cannot produce it to produce it once again. Please understand what an auxotroph as well as a prototroph is. They're listed here on the picture for you, but I would like for you to have at least a small textbook definition because it will definitely make you understand the Ames test. And once again, something mutagenic does not mean it is cancer-causing and carcinogenic, but if it is carcinogenic, it is definitely mutagenic. And the Ames tests for mutagenicity, not carcinogenicity.